Okay, I, right before class, put a new set of slides, which is just a buffed up version of this set of slides on the web, mostly because there is an address where you can go get some extra credit. So thought you might want that handy. So that's in there, as well as a couple minor changes, but if you're good enough to print out the slides in advance, there's not substantive changes to this lecture. And one of the changes, however, is this slide, and I did not mention, and you probably should mention in your method section, that the summed areas were averaged across 3 or 4 different observers. So we had 3 or 4 of you, usually 4, measuring any given bird, and then we averaged across the 4 of you. And what that does is get rid of error helps get rid of error due to different observers. It doesn't get rid of it entirely, but it helps. And I think I talked to the people on Wednesday about my lab partner when I was an undergraduate, Dougie. And Dougie always had hemp flakes for breakfast. And Dougie would get some of the wildest numbers that you could imagine. I was stuck with this guy every darn time. So this kind of gets rid of the Dougie effect. If somebody is getting, wild numbers, I've got 3 more of you. So hopefully you didn't all have hemp flakes for breakfast. And so that kind of dampens down somebody who's like getting kind of wild numbers. Now, what would that do if we were averaging across 3 or 4 observers? What would that do to the variance in our data? Would it make it go down or go up? So we got Dougie, who's out on planet X, and 3 fairly reasonable people. If we just left it alone like that, is the variance large or small? Large. Okay, now we average it. Is the variance going to go down or up? It's going to go down. Okay, so we can kind of keep those outliers from blowing up the variance. All right. And in this lecture, what I'm going to do is I am going to first give you some background on statistics, and then try to walk you through how to write your results section. And the background on statistics will, to a degree, I hope a large degree, help and inform your write-up of the discussion, okay? Because sometimes the pattern of data that you get may have some understanding, or you may be able to understand it better if you think about what's going on with the underlying statistics. Also, in neuroscience, psychobiology, and psychology, and a lot of science, our findings are based on statistics, and so just having a good understanding of statistics is probably one of the most valuable things I can convey to you. So if you can wrap your head around this, it's one of the parts of this course I feel has got the greatest take-home value. Even if you're just reading papers, you can then digest what's going on in the paper. So I am given to understand that at least most of you have some background in statistics, so I'm going to just talk briefly about what statistics do versus parameters. And statistics are based on what? Samples or populations? Samples. And how about parameters? It's a trick question. Samples or populations? Populations. Okay, so it's the job of statistics to estimate the population parameters. And so the mean of the sample, which we would symbolize by x bar, is going to estimate the mean of the population. And the variance of the sample, which is the square of the standard deviation, is going to estimate, oh, that changed back to a Latin character. I'm sorry, that should be a sigma squared, variance of the population. So one of the drafts, that should be a sigma squared, not an s squared, sorry. I'll have to change that. And then people have a lot of trouble in thinking about squared units, okay? So for example, if I were to turn to you and say, how many square feet in this room, what would you tell me? Now give me a number. Actually, that's not bad. Not bad. I once had somebody tell me 4. It's like, that's a room 2 by 2. You know, okay, so I would guess it's actually probably about 900. Looks like about 30 by 30. So, but that's not bad. Not bad. A lot better than 4. Okay, so people have a lot of trouble thinking about squared units. So usually we talk about standard deviations of samples or populations, which is symbolized by sigma. This, by again, shouldn't be s squared. This should be sigma squared. Okay, so just to remind you of that, so statistics estimate parameters. And now we're going to talk about the statistic that we use, which is the analysis of variance, or ANOVA. And it is an F statistic. And it's an F statistic because of the person who invented it, Sir Ronald Fisher. And I'm given to understand that he invented it while at a field station with a slide rule. Anybody ever seen a slide rule? Yeah, you've actually seen one? Anybody heard of a slide rule? Now, before there were 4-function calculators, all of us nerds carried around these big logarithmic scale rules. So if you had to do complicated math, you'd use these things, and they were kind of a pain. But calculators and computers are a lot better. At any rate, he didn't have a lot of high-powered computers behind him. And I'm also given to understand, someday I got to look this up and see if it's true, that in his time it was believed that Mendelian genetics and Darwinian evolution were not compatible stories. And that probably seems bizarre to you, but you know, people were thinking one can be true or the other can be true, but they both can't be true. And he's the guy who figured out how to meld them together. So not such a slouch. Anyway, Fisher's insight was that we could estimate the population variance 2 different ways. And the numerator of this expression is going to estimate the population variance, and the numerator is sensitive to differences among sample means. Okay? And the other way that we could estimate the population variance is in the denominator, and this also estimates the population variance, but it's not sensitive to differences among sample means. Okay, so we're going to have 2 different ways to get to the variance, an estimate of the variance of the population. All right, now the F statistic is an inferential statistic, and so all good inferential statistics come with an hypothesis. And what is the hypothesis? All right, okay. So so it's called the null hypothesis, okay? And let's think about what would the F ratio be if the null hypothesis were true. And the null hypothesis is that all the samples came out of the same population, so they're all estimating the same population mean and also the variance. So the mean of the first population, same as the mean of the second and the mean of the third, on to however many groups you've got, the mean of the nth. Okay? So if that's true and they're all estimating the same thing, what would you expect the F ratio to be? So you can go back a couple. There it is. What would you think it would be true? If it were true, what's the F ratio going to be? Say it loud. 1. All right, everyone see? Okay, so you got 2 estimates of variance. If there's no difference due to means, they should be at the same number. We should end up with 1. Okay, so I'm the kind of guy that I got to see a picture before I can really understand it. So to get to the numerator of that expression, we're going to estimate the variance of the population by finding the variance of the sample means around the grand mean. So we're going to put the grand mean in there. And the grand mean is if you take all of the individuals, no matter what group they came out of, you stick them in a big pile, you get the average, okay? And that's going to be the best estimate that you can come up with of the population mean, right? Say yeah. Yeah, all right. Okay, and that's if the null hypothesis is true, right? Okay, so we're going to assume that. Okay, so what we can do then is we can say, oh, all right, so I have all these little distributions here, and all these little distributions are representing, say, 3 different groups. I didn't want to stick in 5 groups like we have because that would be kind of visually noisy. All right. So what we could do is we could figure out the deviation of each of our little sample means away from this grand mean, right? Okay? And we could use that to estimate the population mean. And the way I like to think about it, what we're basically doing here is figuring out the standard error of the mean. Standard error of the mean is how much on average our sample means would deviate away from the population mean. Want me to say that again in slow motion? Okay, standard error of the mean is really a measure of how much on average our sample means would deviate from the population mean. So we actually calculated this for our groups in lab, and you may remember that the standard error of the mean is equal to the standard deviation over the square root of n. So if you are essentially finding the standard error of the mean by finding out how much these sample means vary from this grand mean, you're going to be able to get to the variance of an estimate of the variance of the population, right? Because that's just the square of the standard deviation. That's a couple of simple math moves. Right? Okay. All right. So that was the numerator. Okay. So we can also find the variance, an estimate of the population variance by finding the variance within each sample. So we take each sample, and so in this first case, I'm going to be taking this first little sample, and I'm only going to show you 3 points, but we'll say, okay, so how much do all the points in this first sample deviate from or vary from their mean? And then we go and we do it for the second sample, same thing, figure out the variance for the second within the second sample, and then the third group, figure out the variance for the third group, and then we can average across all of those. All right? So now we have 2 different measures, 2 different estimates of the population variance. One depended on where the sample means were, one didn't. Okay? That's how it works. All right? So then we come back and we can estimate this. And as you already told me, if the null hypothesis is true, it should equal 1. All right? And this statistic, this is what the data might look like if the null hypothesis isn't true. So you might have at least one group that's way the heck away from the others. All right? Okay, when the null hypothesis is true, you can figure out what the distribution of this statistic should look like. And depending on the sample size, it should look like one of these. Ours is going to be actually closer to this kind of yellow distribution, or maybe this one that says 5 and 10. Something like that. So the point is, if the null hypothesis is true, most of the time your F ratio is going to be around 1, right? Okay. And you could then figure out a point that cuts off 5% of the distribution. So you can get out your old calculus book, right, and integrate under the curve and all that great fun, right? Yeah, all right. Now the good news is somebody for all conceivable sample sizes already did all that work for you and made tables, okay, and then stuck them into a computer so you don't have to do all that nasty calculus, all right? But it makes sense to you then if I say, all right, we can then have some cut point where there'll be 5% of this distribution out in this tail, right? Okay, so what if we get an F out there in this tail? So we get an F of, say, 25, as we tended to in our data. What are you going to say about it? Is the null hypothesis true? Yes or no? No. Okay, that would be a very unlikely outcome. Okay. On this scale, an F of about 25 would be over there in Franz Hall somewhere. Okay, so that's an extremely unlikely outcome. It's no longer reasonable to cling to that null hypothesis. So then we say, okay, null hypothesis isn't true. And we did get a significant difference among our sample means. Now, are we going to know where that difference is? No. So the F ratio stops there. It just says there's a difference in there somewhere. You know, if you get a significant F, but doesn't tell you where. Okay, let's talk about some reasons why one would perform an analysis of variance as your statistic. Okay, so one of the reasons that one would use this statistic in ANOVA is you, rather than do things like multiple t-tests, and back in the day, let me tell you, running an ANOVA was a huge pain. You guys get to do it in about 500 milliseconds, but as I told the Wednesday people, it used to take me all afternoon and a whole six-pack of beer to do these darn things. The beer did not help, by the way. And a t-test would take half an hour, all right? Now that's not a big consideration, but believe me, we did not like to have to do them because they took forever. So I always say, why don't we just do a whole pile of t-tests because it's a lot easier. All right, so one of the reasons for doing an ANOVA rather than a whole pile of t-tests is you can analyze for an interaction when you have more than one independent variable, that is to say a factor. Okay. Now, is that going to be a reason in our study? No. How many factors did we have, independent variables? We only had one, but if we had, say, a different study that we were looking at the effects of a drug across males versus females, then you have, say, two variables. You'd have sex and drug, okay? And you could see if males and females acted the same or something like that. That's when you're looking for an interaction. Doesn't come into play in our study because we aren't looking for interactions. Let's talk about some things that do come into play, okay? One of the things, one of the big reasons why you would do an ANOVA rather than a whole pile of t-tests is that it protects against experiment-wide alpha error. Okay, and an alpha error is also known as a type 1 error, or you could also think of it as a false positive. Okay, it's your false positive rate. So it's when you're going to say there's a difference when in reality there was not. All right? And so I'm given to understand, at least some of you took statistics, so what is the probability generally of an alpha or type 1 error? If you do one t-test, say. You all did t-tests, right? Yeah, okay, do one. Okay, it's comparison of two means, all right, you only have two groups. Do one t-test. What's the alpha level? Oh, it's good we have this conversation. Okay, so your false positive rate is 0.05, okay? And we know that, we're going to live with it, okay? That's part of the game. Okay, that's if you do one. Now, if we were to do t-tests for all of our different groups, let's see if I can figure this out, we would have, let's see, you'd have 4 comparisons there, plus 3, it would be 7, plus 2, plus 1. So I think you'd have 10 separate comparisons if I did my math right, okay? Is that right, David? Yep, okay. So you'd have 10 separate t-tests you'd have to do to compare all the groups together, all right? So what happens? So you do one t-test, you have a false positive rate of 0.05. What happens if you do two t-tests? What's your false positive rate? Close to 0.1. It's not quite linear, but in that range, actually, it gets close. So you do 3, you get about 0.15. You get up to about 10, and you're close to about 0.5. Not quite, but you're close. Okay, so that's a 50% false positive rate. Ooh, that's a little bit high, and we don't like that in science. So if we do just a whole pile of t-tests, that's what we're going to end up with. We're going to have a good chance of false positive. We don't want to do that. So what the analysis of variance does is it makes all the comparisons at once at an alpha level or a type 1 error or a false positive rate, if you will, of 0.05. Okay, so it keeps that from inflating. And in our kind of study, that's important. Okay, so all the comparisons are made at 0.05. Okay, so that's really good. And then the other thing that the ANOVA will allow is, since we can pool all of our subjects together in the analysis, we get more statistical power. Okay, and is that a concept with which you are familiar? No. Okay, well, let's talk about that because it's important. Statistical power is the probability of rejecting the null hypothesis when it's false. Okay, so in other words, it's the probability of getting a true positive, or another way to say that is a correct rejection. Okay, so this is detecting a difference when it's really there. Okay, so do we want this to happen? Oh yeah, you want to see the differences when they're really there. What I'm going to do now is talk about some factors that influence power, and some of what I'm going to say may be relevant to interpreting your data, especially when your data doesn't match my data that's published. Okay, and correct me if I'm wrong. You Tuesday people, any Tuesday people present? Okay. Did you ever get 15 micrograms being significantly masculinizing? Do you remember? Never? Well, my memory is it rarely or never happened. Okay. It was close. Yeah, it was like on the border, but was kind of on the wrong side of the border, right? Okay, so especially you guys, some of this is going to matter to your interpretations. I think the Wednesday people, you got 15 to work sometimes at least, right? Okay. By the way, your data is not the same data Tuesday to Wednesday, so don't go flippy-flopping, okay? Because you're going to mess up your TAs, and that's not good. All right, so let's talk about some factors that influence power, okay? And what I'm going to here represent is in this blue distribution, say this is the set of sample means that you would get if the null hypothesis were true, okay? And of course, and then we're going to have this green bar, and this green bar is going to cut off, let's say, 5% in this tail of this blue distribution where the null hypothesis is true, okay? So that's our what kind of error? If we're cutting off 5%, and the null hypothesis is true, if it's to the right of that green bar, we say, yes, there's an effect, even though it's actually coming out of that blue distribution. So is that an error? Yes, so null hypothesis is true. There's no difference among the means, okay, or between the means, okay? So if we are actually pulling a sample out of that HO distribution and we say, yep, there's an effect, we made a mistake, right? We can't know that, but we can know the probability that we're going to do that, okay? And that's how much? 5%, okay, your alpha level. Does that make sense, or am I confusing y'all? You're not looking good there. Okay. Just pay attention to the blue distribution, okay? Got a green criterion line there, all right? Any outcomes to the right of that green criterion line, we're going to say null hypothesis is false, okay? Let's say we only have that blue distribution. The null hypothesis is, in reality, true. We say it's false, okay? And so that's our type 1 error. That's a mistake. We got to live with it. I hate to break this to you at such a young age, but it's not a perfect world. All right, so we can make mistakes, okay, and we know that, and that's going to happen 5% of the time. We just live with that in science. Okay, Now consider the red distribution. All right? That's when the null hypothesis is false. There are differences. Okay? And that's the frequency of outcomes as a function of some variable, or let's say t value or something like that. And so everything from that green criterion line to the right will reject all those outcomes. All right? So those are all going to be our true positives. That's going to be our power, right? But we're going to miss some of those outcomes. We're going to miss, in this red distribution, we'll miss all these outcomes here, right? Okay. So that's a different kind of error. That's a type 2 or beta error, which you may have heard of. Hear about these in statistics? Okay. Glad we're having these talks. All right. So so we don't want type 2 errors either. These are when we should reject the null hypothesis and we're not. Okay, does that make sense? And so the null hypothesis really here in all of this region is false here for this red curve, but we can't tell that, and so we don't reject the null hypothesis. And that's a mistake too. So if the difference between the means goes up, then we're going to be able to shift. So you can think of when you have an effect due to something that you're taking your two distributions and you're just adding a constant to every value, okay? So that constant can be small if you have a small effect, like here in this top panel, or it could be large if you had a big effect, okay? And what that's going to mean here with this green criterion line is now you're going to be able to reject all these outcomes in this second distribution, right? All right? Whereas you couldn't do that when you had a smaller difference between your overall means. Does that make sense? So, big effect sizes, your power is going to go up. All right, that's the bottom line. So, if you're designing an experiment, doesn't matter in what, don't do something subtle. Okay? You do something subtle, you're going to have some situation like this where you hardly move those two distributions at all. So as one of my mentors told me, hit it with a sledgehammer. Don't do something subtle. If you want to see impacts, hit it with something big. You can go back and look for subtle later. But if you start with subtle, you don't know, and you get nothing, you don't know. Well, did it not work, or was the effect size just so tiny I couldn't detect it? All right. So you're starting out, hit it with a sledgehammer. All right. Especially in our state of knowledge now, you don't want to really look for subtle. We need to find out big important things still. We're still at that level. All right. So effect size. How might this relate to your data? So you guys with the 15 versus 0, okay, and versus 50 versus 0. So the 15 versus 0, you might be more like this, where you didn't shift it very much, okay? Your effect size was small relative to, say, 50, all right? And so that could influence whether or not you get a significant difference. Okay, does that make sense? Okay. All right. Another factor that influences power is the sample size, okay? As the sample size goes up, power goes up. And the reason for that is these distributions skinny up as you get a bigger sample size. Both your null hypothesis and your alternate hypothesis distributions skinny up. And the reason for that is as you get bigger and bigger sample sizes, you have better and better and better characterized the population mean. So let me give you an example. So it's useful to think of it at the limits. So let's say you wanted to figure out the distribution of heights of American men. All right, you're going to do now a sample size of 1. You reach into the population and you randomly grab out Dr. Grisham, a stunning 5'4". All right, so you go, okay, there's our estimate of the mean. There's Mean man is about 5'4". Okay, so so that's a very small sample. Small samples are crappy estimates of the population mean. Okay, so let's say we had the whole population. It's useful to think of it at the limits, and we left out. Is Yao Ming an American yet? No? Okay, well, give me a basketball player. Shaq. Okay, we leave out Shaq. We got everybody but Shaq. All right, we find the mean. Okay, is that going to be a good estimate? Got everybody in the whole bloody population except for Shaq. All right, even if we leave Shaq out, that's our biggest possible sample is everybody except for one. Even if we leave out that big outlier, we're going to have a pretty good estimate. Okay, so the bigger your sample size, the less variable these things are going to be. So these distributions skinny up, and the net result is you're going to be able to reject more and more and more of these instances where the null hypothesis is false. All right, so that's why you want good size samples if you can manage it. Of course, it's always more work. Okay, another factor that influences power is the variance. As the variance goes up, power goes down. Okay, and so what I'm going to represent the variance in scores, what that means is these distributions get fatter. All right, and so now we have to determine our green cut line based on the HO distribution. I'm going to hope that's 5%. And so now you see in this bottom panel, we're only rejecting a few of these HA instances, right? Versus when the variance is less, we get to reject a larger proportion. So let's talk about what that's going to mean for, say, your data. So when I measure things, or when we measured things for that paper, I had one undergraduate per dependent variable, all right? Whereas you guys have got several people per dependent variable, right? Okay, so that means that your variance relative to mine is going to be high or low? High, because everybody does it a little bit differently, all right? So relative to the data that we published, your variability in scores is going to be higher, all right? So what's that going to mean? Are you going to get fewer or more instances where you reject the null hypothesis? Fewer, okay. So there may be instances in your data where we find significant differences, like we got a significant effect of 15 micrograms, whereas you may miss it in your data. And again, I think I'm addressing mostly the Tuesday people. I didn't check over the Wednesday carefully. But if your data doesn't match mine, that's one of the very likely reasons. Okay, you got lots more variability due to multiple observers. That's just the way it goes. Okay, and just for sake of completeness, I'm going to talk about this last factor that influences power. We don't get to play with it very much, and that's the alpha level. And by convention, in biology and in psychology and in science, the alpha level is pretty much set at 0.5. Okay, if you are in an instance where you're doing a study and you say this drug may be toxic and you want to make sure it's not, you can bump up the alpha level. Say make it 0.10 or even 0.25 to make sure. All right, that's not generally how we do it in science, but if you have some compelling reason like that, you might want to do that. Okay, and then so what you're doing essentially is just shifting this criterion to a more lax standard, and so then you'd be able to find more of the instances and reject them. Okay, but again, in science, we don't get that luxury. All right, so now, after I've done all that, let's talk about some of the nitty-gritty and how to write up a results section. So the first thing you're going to want to open with is talking about your overall F. And you have 3 overall Fs, right, on 3 dependent variables. And I think it would make sense to have at least, say, 3 short paragraphs, one for each of your dependent variables. So please don't copy this sentence. And I made up some numbers here. Actually, I said it was RA. It's something else in your data, but so don't trust this. This is not your, did I write that? Not your data? There it is. Okay, this is not your data. All right. So, you know, the first thing you might want to say is you got something like significant differences are found between or among. Would you say between or among groups? A little English lesson. If it's two, it's between. If it's more than two, it's among. All right? That's how you know if you're well educated or not. Okay? So don't make that mistake. All right. We're found among groups in the analysis of RA sums, because the RA sums is actually what we got by way of a dependent variable, right? And so you're going to make some statement like that, similar to that, in words for each of your dependent variables. And then what you need to do is follow that up with some numbers, okay, that solidify what you've said in words. And your numbers should be consistent with what you say in words. So, this is actually the right way for reporting an F. And so, and that's what you get out of ANOVA. Okay, so it's F, and in our case, Fs, by the way, I'm not going to go into this because I don't have time. Fs have a degrees of freedom for the numerator and degrees of freedom for the denominator. So how you get the numerator, you go to your dependent measure, and the degrees of freedom are the number of groups minus 1, so 4. And then the denominator is the error. And so that's actually the number of subjects minus the number of groups, so 25. So I believe in all of your instances you have 4 and 25 degrees of freedom. That tells the reader how many groups and how many subjects, if you know something about F statistics. Okay, then you should give them the value, which we got here, and then you should make some probability statement about getting that value, which you're going to get here in this sig column. So you should say P, in this case, less than 0.001. Please don't say that the probability is equal to 0. That means it could never happen, and that's not true. Okay? So we know it's less than 0.001. Yeah? It's just off whatever. I don't know what it's doing. It's probably, it has a lookup table, and that's where the lookup table ended, so it just kicks out zeros. We know it's at least less than that. I suspect it's probably 0.00. I would bet it's 4 zeros and a 1, but I don't know. I didn't look it up. Okay, important point is this deadly little less than sign, okay? Make sure it's going in the right direction, all right? If you flip this around, you just contradicted what you said in words, okay? So make sure on that one that it's P less than if it's significant, okay? If it's not significant, it's going to be P greater than 0.05, which I don't think you need to talk about. But make sure that that little less than sign goes towards, it's pointing towards the P, okay? All right, because that's a frequent and deadly error. All right. What at some point in this discussion, I would refer the reader to a graph, okay? You can say, you know, see figure 1, 2, or 3, whatever. You should have 3 figures, right? Refer them to the graph. And what you're basically then going to do in the text is walk them through your graph, okay? Verbally walk them through your graph. And by that, I don't mean say the mean of the males was this and yada. Don't do that. And also, please, I beg you, don't put the values of the means by the various points, because that kind of implies that we're not smart enough to be able to read a graph, okay? It's just redundant if you do that, so please don't. Also, while I'm at it, I hate the Microsoft gray lines on gray background. Let's camouflage our data so no one can really see it. Don't do that. Make it pretty, okay? So it's easy to see. All right, so you're going to walk your reader through the graph, and when you do that, make reference. Okay, by the way, units, units, units. Either put units on your graph or put it in the figure caption, okay? Don't ignore it. So this is not a perfect graph. I should have put units here. I like it because it's easy to see and the labels are big. But, okay. So then you're going to, as I said, you're going to walk your reader through your graph. Make reference to your post hoc tests, okay? So one thing you might want to say is the males were significantly different from all other groups, which they were, right? I think in every last measure that we did in both classes. And then you want to, in parentheses, say Fisher's post hoc test, Fisher's LSD test, P less than whatever you got there, whatever your minimum is, okay? Want me to say that again in slow motion? All right. So you want to say, you know, as you're walking your reader through the graph, you want to say the males were significantly different from all other groups on this measure. Fisher's LSD test, all P's less than point, say, 0.001, okay? So use your post hoc tests to back up what you're saying in words. Okay, and you should do that essentially and then tell them about if you've got these points, you know, the 50 and 15 different from these points, you should say back that up with your Fisher, your post hoc tests, okay? What are your probabilities that show you that? Okay, back it up. Okay. And again, all right. You got 3 dependent variables. You're going to have 3 graphs, 3 sets of analyses, and it sounds like you ought to have about 3 paragraphs. Does that make sense? Okay. All right. We went over some of these points last time, but they probably bear repeating. So then, after you get done with your results section, you want to go into your discussion. What you're going to do in your discussion is kind of the opposite of what you did in your introduction. Introduction, you went from the broad down to the narrow. You're going to reverse course in the discussion, go from the data to talking about especially answering your questions, okay? So we're going to use the data. Go back. It's good to refer the reader to figures in the discussion also to make your points, okay? So they can go back and say, oh yeah, that was true about that, okay? So if you're going to talk about something in your data, tell the reader what graph to go look at so it makes sense to them. All right, so first thing we want to know, we wanted to see if we could replicate the sex differences, right? Okay, and we're going to play, this is not your data, but we're going to play like it is your data. So what would you say, gang? Did we replicate the sex difference? Yes? No? Why? Why would you say this? Who is different from whom? Males are different from everybody else, and especially the 0 females, okay? So right there, that gives you your sex difference. Is that good? Yeah, okay, because people criticize biologists for replicating things unnecessarily, but if you don't replicate it, then there's no reason to go and explore the phenomenon further if you can't get it. All right, so you got to establish you can really get it. So you guys did a great job. You all got that. That was great. Okay. So yes, we replicated the sex differences. You should tell the reader what aspect of your data allows for that conclusion. Did you get it in all the variables or not? And does it replicate things in the literature? If it did, reference them. You all read the stuff yet? One of you. Anyway, there's some good papers there in your reader, and you might see if they also got sex differences in the song system. If they did, say, tell us about it, and reference them. All right, one of the big motivations for this study was to gauge the sensitivity of the female zebrafinch song system to estradiol. And we wanted to know, is the system exquisitely sensitive to estradiol? What data allow your conclusion? And does this say anything about the sensitivity of the song system in males? So let's talk about it. So what do you think? If you get data like this, which is also not your data, if you get data like this, what do you think? Is the song system exquisitely sensitive to estradiol? No, why not? Yeah, 5 is the same as 0, didn't set it off, didn't masculinize it. So, and 5 actually, in other systems, does a lot. You can, if you gave it to a baby rat, baby female rat, she would never have ovulatory cycles in her life. So it would really masculinize her brain. Okay, so you talk about the data. Again, if you find it in more than one of your dependent variables, you should talk about it. Does this say anything about the sensitivity of the song system in males to estradiol? Not really. Could be completely different, right? Okay, we wanted to see if masculinization is an all or nothing event once the threshold is reached, or if it's a graded response. And we also talked about this last time. What do you think, gang? Is it graded, or does it look like a threshold? Okay, so if it's graded, that means that if you give a little bit, you get slight masculinization, but significant masculinization. And if you get a little more, you get a little more masculinization. You give more, you get more masculinization. So you would expect these points to be different from this point and different from each other. Okay, if that were true. So what do you think? True? All right. I'm going to leave you with that. Okay. We wanted to see if the different parts of the song system show differential sensitivity to estradiol. Okay, so these are actually not your data. These are my data. That's why there are no males. And I got data like this. Okay, so does this data give you any evidence of differential sensitivity? Okay, if it were differentially sensitive, what do you think you'd see? So let's say the X, if it were differentially sensitive, 5 might masculinize X but not the others, okay, or something like that. But if it's the same doses show masculinization and the other doses don't, that's not differential sensitivity, right? Okay, and then the last thing was we wanted to see if estradiol will fully masculinize the song system in females. Okay, gang, so let's say you got data that look like this. Yours is not going to be far off that. What do you think? Didn't work to do that, right? Okay. Lastly, you're going to want a concluding paragraph, talk about any differences or similarities to Grisham et al. If there are different patterns in your data relative to the Grisham et al., why is it different? Any reasons for that? Okay. And you want to summarize. You want to kind of tell them what you told them, that don't be redundant. Don't go back and start reporting means and stuff like that. Please, please. Do a nice summary. Don't be redundant. Okay? All right, you're going to need a reference list. I'm going to ask you to do it in APA style, which I think there is a link on the course website. And I'm going to ask you to go find one reference that I haven't given you, using PubMed or something, on the song system, and use it in your write-up and attach a copy to your lab report so your TA can see if you know what you're talking about. All right? And then lastly, I'm going to put on an addendum, put your data and conclusions of the sex of the mystery animal and why you got your conclusions after the references. And I'm going to give you a picture. Actually, I have given you a picture. You're going to give me the picture you took, and somebody on Wednesday took this beautiful picture of an RA. So yes, you all can do this kind of work, I know it. So there's an RA. It's either male or female. You get to choose and tell me why you think's that. Okay? All right. If you'll give me a couple more minutes, I'll tell you how to go get some extra credit. All right. Want some extra credit? You can go to this website, and this will be part of a for real study. We are actually have funding from the National Science Foundation to give away these teaching tools to the world. And so what we're trying to see is, what did you learn from this? Okay, so there's a little tiny quiz that I'd like you to take. That's the student post-test. And it doesn't matter what you score on this, you'll still get 1 point extra credit for taking this. And there's also a materials evaluation where you can give your opinions about the materials and stuff that we used in this module. And we'd really appreciate the feedback, because we're still tuning this up. It was the first time it was offered in this course. And again, we're trying to give it away to the world and make it really an excellent module. So your feedback is really valuable to us. Since this is a for real study, I am obliged to tell you, if you want the extra credit but you don't want to participate, you can approach me about an alternate assignment. Okay? I have to make that option available. You can't get extra credit and extra credit. You can do one or the other. All right, and I did have planned to tell you a little bit about hormones in humans, but I've run out of time. So if you want to leave, feel free. You want to hear the story? Okay, I'll tell you. Actually, I want you to hear this, so feel free to pack up. Here's how it works with mammals. And most of the time, it's steroid hormones that are responsible for organizing sex differences in the central nervous system. And in the periphery, so I mean the body and the spinal cord, it's androgens. No doubt about that. And that seems to be true in humans. There are some minor exceptions, but not worth talking about. And this is organizational effects, okay, not activational effects. As far as masculinizing the brain in rodents, no doubt about it, estradiol. In humans, I went through tons of literature, and what I looked at was sexual orientation, and it doesn't look like estradiol is responsible for masculinizing brains in humans. That's from just surveying a whole lot of literature. It may be androgens in humans. So the point is, it behooves us to keep looking at various models of sexual differentiation to figure out what's ultimately going to be true of us. And that's where I got to leave it. It's kind of a messy story right now, but that's science. So thank you for your kind attention.